Today we are going to study the secondary structure in the stem of sunflower. So secondary growth is the typical pattern in the dicot plant where we can find because of the secondary growth there is a growth in the girth as well as in the height. So today we are going to discuss on the same topic, how the plant grows by their secondary growth. Actually, the secondary growth is nothing but the formation of secondary tissues in the lateral meristems. Now we understand the meristem. Meristem is nothing but the dividing tissue. It increases the diameter of the stem. Diameter म्हणजे girth of the plant किंवा ज्याला आपण झाडाची रुंदी म्हणतो. So it is the increasing in the diameter of the stem in particularly woody plants. So secondary tissue which constitutes the bulk of the plant. This take part in the providing protection, support and conduction of water and the nutrients. कारण जशी झाडाची उंची आणि रुंदी वाढत जाईल, it needs a more food and support, so it is provided by the secondary tissue development in the plants. The secondary tissues are formed by two types of the lateral meristems. One can call the vascular cambium and another one is the phellogen. So cambium or phellogen and the vascular cambium, these are the continuously dividing tissues in the plant which gives a secondary structure, secondary tissue development in the plant. The vascular cambium produces secondary vascular tissue with the phellogen and the phelloderm tissues. So here you can observe that how the secondary growth has been taken place here. The uppermost layer, as we call it as a epidermis, then the second zone which is followed by the epidermis is normally a collenchymatous hypodermis tissue. Then we go for the parenchymatous cortex. Cortex is followed by the endodermis, and this endodermis is normally followed by the pericycle. Now this tissue, this cortex, if we left that cortex, we go the secondary growth. Here you can find there is the development of the vascular bundle along with the cambium, metaxylem elements, protoxylem elements and central pith. When these primary structures, particularly vascular bundles, when they are multiply grows, then we call it as a secondary growth. The formation of secondary vascular tissue, they form by due to the vascular cambium. Now vascular cambium is nothing but the producing xylem towards the center and the phloem towards the periphery. So likewise we can continue with that. The vascular cambium is produced fascicular and interfascicular cambium. So this vascular cambium is modified into various ways itself, and at various levels it starts developing the tissue. The interfascicular cambium is particularly a meristem. Now this meristematic tissue is, as we know, it can able to produce phloem tissue as well as the xylem tissue. So interfascicular cambium is primarily the meristem which occurs at the strips in the vascular bundle. Vascular बंडलच्या आतमध्ये ज्या स्ट्रिप्स असतात तर त्या स्ट्रिप्स ज्या आहेत, here you can observe that. So these are the strips which are nothing but the cambial elements. So these are the interfascicular and which is arises secondarily from the cells of medullary rays which occurs at the level of interfascicular strips. The interfascicular strips ज्या आहेत त्या ह्या आहेत. These are the medullary rays where we can find there is the interruption of these cells has been taken place, or the cambial cells has been taken place. That can be found in the younger stem where we can find the primary development is there. At secondary level, the tissues has been get modified and the secondary xylem, secondary phloem and the all secondary tissues like the development of the cork cambium, phloem and the phelloderm. So all these tissue has been started here. These are the intermittent medullary rays can be observed here. All these elements, these are the secondary xylem elements and these are the medullary, where we can find here the primary xylem only in the center. Otherwise, the all secondary tissue has been started developing here. Secondary cortex is there and secondary phloem, xylem, all the elements are found in the secondary growth of the sunflower stem. The cells of the vascular cambium are of two types basically. Either they are fusiform initials or they can be the isodiametric ray initials. Here you can find that. Here we can observe that the diagrammatic sketch of that particular interfascicular cambium. So this ring is nothing but the interfascicular cambial ring. So here you can find these are two types of the fusiform initials and short isodiametric ray initials can be developed here. Then fusiform initials divide to form the secondary phloem. Fusiform initials चं काम काय आहे? It gives the secondary phloem on the outer side of the secondary xylem on the inner side. आता inner side ला काय आहे? Always there is the development of the xylem, and whereas the outer side of the cambium or towards the periphery of the tissue stem, we find there is the development of secondary phloem. With the formation of secondary xylem towards inner side, the vascular cambium moves gradually outside, adding a new cells. The fusiform initials, which gives a fusiform cambium cells, gives the phloem cells, which are developing outside the cambial ring. Then cambial cells and xylem mother cell. Xylem mother cell is towards always inner side of the tissue. Here cambial cell produces outer side, which is the phloem, and the inner side towards the xylem. So this is the typical development of the cambium. Fusiform cambium पासून बाहेरच्या बाजूला काय तयार होणार आहे? फ्लोएम तयार होणार आहे आणि आतल्या बाजूला झायलेम तयार होणार आहे. तर फ्लोएम मदर सेल आणि झायलेम मदर सेल and cambial cell. Likewise, there is the division of the cells has been taken place. The phenomenon is called the dilation. Now these cells, the new ray initials are also added gradually and they can form the ray initials every year. कारण secondary growth कुठे आहे आता? जिथं perennial plants आहेत. Those plants are perennial. Only in those plants we can observe the secondary growth in the plants. Those are annuals, म्हणजे त्यांची लाईफ सायकल एकाच वर्षामध्ये पूर्ण होणार आहे. तिथं फक्त आपल्याला प्रायमरी ग्रोथ डेव्हलप झालेली दिसेल. But the multiple rings or the multiple tissue has been developed in the perennial plants, and where we can find the multiple vascular cambium can be developed there. Where the vascular cambium undergoes the divisions, and these divisions are multiplied anticlinal or for the periclinal divisions has been taken place, and these divisions gives rise to the tissue towards the inner side as well as the outer side. Here in diagram you can find that. So this is the diagrammatic sketch for that, where the central there is the pith. These are nothing but the pericycle layer. Pericycle च्या बाहेरच्या बाजूला सगळा cortex, phelloderm आणि periderm develop होणार आहे. This is the innermost ring of that is the pericycle. मग pericycle मध्येच आपल्याला इथं काय डेव्हलप झालेले दिसत आहे? These are the primary phloem patches. आता हे प्रायमरी फ्लोएम पॅचेस आणि हा सेकंडरी टिश्यू इथं आता आपल्याला मॉडिफाय झालेलं दिसतं. Here you can find these are the primary phloem patches going gradually outer side, and here at the center in the inner side there is the xylem elements, and this complete ring is of the secondary phloem. So secondary phloem is developed in the form of a ring. So secondary phloem and secondary xylem is developed in the secondary growth of the xylem elements. There are various modifications of the tissue is being taken place. So there is the development of vascular rays. What are these vascular rays? These are nothing but the secondary medullary rays are radially arranged cells. In last lecture we discussed about that. In two vascular bundle, whatever the space is there, that is occupied by the medullary rays. So these are the medullary rays, here marked as this circle. These two are the vascular bundles. हे vascular bundles आहेत आणि त्याच्यामधून हा जो गॅप इथं दाखवलेला आहे, this is a medullary ray. It is in between the two vascular bundles. There are a few cells in height depending upon their breadth. The vascular rays are mostly uniseriate. ते फक्त एका सिंगल रो मध्ये तुम्हाला इथे दिसत आहेत. ठीक आहे. medullary rays are normally in the single row-like structure, and they are mostly uniseriate, or sometimes we find the multiseriate. In last lecture, we discussed about the multiseriate type of the rays. These cells are nothing but the vascular rays which enclose intercellular spaces. आता इथं तुम्हाला दिसत असेल, here you can observe that whatever little bit gap is there, that gap is filled by these rays. इथून हे इथपर्यंत इथं कंटिन्युअस झालेला आपल्याला दिसतो. So these medullary rays are placed in between the two vascular bundles along with the intercellular spaces. The vascular rays conduct water and organic food material and also permits the diffusion of gases in the radial direction. काल आपण बघितलेलं होतं की वेसल एलिमेंट्स किंवा ट्रकीडचं काम काय आहे? दे आर नॉर्मली फ्लो द न्यूट्रिएंट और वॉटर फ्रॉम द वर्टिकल डायरेक्शन फ्रॉम रूट टू दी लीफ. बट वेनेवर देअर इज रेडियल डायरेक्शन म्हणजे त्याच्या गर्थ मध्ये जेव्हा ट्रान्सफ्युजन व्हायचं असेल, सो ऍट दॅट टाइम these rays will allow these water and minerals to transfuse by the radial direction. आणि म्हणून त्याचा इथं महत्वाचा रोल आपल्याला बघायला मिळतो. Then what is secondary phloem? जसं आपण secondary phloem मध्ये काय होणार आहे? These are normally the again parenchymatous cells and forms a circle outside of the vascular cambium. त्याची एक रिंग तिथे डेव्हलप होणार आहे. This secondary phloem does not grow in thickness because primary and the older secondary phloem is present on the outer side, gets crushed with the development of the new functional phloem. आणि म्हणून काय होतं, secondary phloem does not grow in the thickness. It is just forming a ring like structure. आणि तो कंटिन्युअस त्याच्यामध्ये डिवाइड होणार आहे. तो जर तिथे डिवाइड झाला नाही तर आपल्याला माहित आहे, आपण मागच्या स्लाइड मध्ये बघितलेलं आहे, देअर आर द पॅचेस ऑफ द प्रायमरी. ऑलरेडी प्रायमरी फ्लोएम इज देअर, or it can be get crushed due to the development of the new phloem cells. Therefore, the rings, the annual rings, normally we call them as are not produced in the secondary stem. Secondary phloem is made up of the same type of the cells that are found in the primary phloem. म्हणजे त्याच्यामध्ये मेटाफ्लोएम आणि sieve ट्यूब जे असतात. So on the basis of that, here also there is the development of the phloem is taken place. Near about the same type of the development in primary and secondary phloem is there. Here we have shown that how the phloem cells has been get modified. These are the bast fibers. They are just gradually one by one adding their tissue. It is not the complete division of that particular cell. It is the gradual development and adding of the tissues or fibers in that. So likewise, gradual development of the secondary phloem or the bast elements has been taken place. It is secondary xylem, developing towards the center as usual, towards the center, towards the pith. Secondary xylem, it forms a bulk of the stem and it is commonly called wood. जे मोठं झाड असतं, झाडामध्ये जो लाकूड ज्याला आपण भाग म्हणतो, so that wood part is nothing but the all xylem elements. These are all secondary xylem elements. This secondary xylem consists of normally vessels, tracheids. They may be the tracheids elements, wood fibers, and wood parenchyma. All these structures are found in the secondary xylem, which helps in the developing wood structure in the plant. This wood parenchyma may contain the tannin and crystals beside the storing food. Stem मध्ये काय असतं, because it is harder in nature, it may store the food, oils, or we can say the another storing material, another storing chemicals. And axial parenchyma, the cells arranged where the longitudinally radial cells parenchyma जे आहेत, they are normally horizontal in fashion. So wood parenchyma which helps in the storing food material as well as oils and resins in that. The latter part is of the vascular ray present in the secondary xylem. आता इथं आपल्याला जो आपण घेतलेला आहे LS of the vascular cambium इथं दाखवलेला आहे. These are all the ray initials. आता LS आणि TS याच्यामध्ये बेसिक फरक काय? LS is nothing but the vertical section and TS is nothing but the ट्रान्सवर्स, ज्याला आपण आडवा सेक्शन म्हणू. सो नॉर्मली फॉर प्रॅक्टिकल पर्पज वी टेक द transverse section of the stem and leaf. Here we can find this is the transverse section. Here it is actual section where we can find there is the development of the secondary xylem and the secondary phloem elements. Here you can find this particular ring, greenish ring, is of the vascular cambium ring. Vascular cambium cha towards center we find there is the development of xylem, and towards periphery there is the development of secondary phloem or the primary phloem or phloem fibers. So here this green ring is the cambium ring. Inner side, this is the secondary xylem tissue is developed. Secondary xylem हा जो intermittent ज्या conditions आपल्या इथं बघायला दिसतात, single cell structures, these are nothing but the xylem rays and these are the xylem tissues. जे छोटे एलिमेंट्स असतात त्याला आपण प्रोटोझायलम एलिमेंट म्हटलेले आहे, ज्या मोठ्या सेल्स आहेत त्याला आपण मेटाझायलम एलिमेंट्स म्हटलेले आहे. सो इट इज द प्रायमरी अँड सेकंडरी झायलेम टिश्यू ऍक्च्युअली इन द सनफ्लावर स्टेम. नाउ दिस इज द सेकंडरी डेव्हलपमेंट वेअर वी कॅन फाइंड देर इज द डेव्हलपमेंट ऑफ अॅन्युअल रिंग्स. तर हे अॅन्युअल रिंग्स कशा डेव्हलप होतात? ऑन द बेसिस ऑफ अॅन्युअल रिंग्स वी कॅन कॅल्क्युलेट द एज ऑफ दॅट पर्टिक्युलर प्लांट. ठीक आहे. मग आता अॅन्युअल रिंग ह्या कशामुळे डेव्हलप होतात? इट इज द वुड फॉर्म इन अ सिंगल इयर. प्लांट्स आर नॉर्मली पेरेनियल, दोज आर ट्रीज. These trees are the perennial in nature. So whatever wood is formed in a single year, it forms a ring. So it consists two types of the woods, particularly spring wood and the atom wood. The spring wood and atom wood, they are soft and hard tissue developed in that particular year. The spring wood or early wood is nothing but the wider than the atom or late wood. म्हणजे जो मोठा भाग आपल्याला इथं बघायला मिळतो that is of the spring wood, and which is one is the lighter one. Here you can find early wood. आणि आता इथं अर्ली वूड आपण बघितलेला आहे. अर्ली वूड अँड लॅटर वूड असे दोन प्रकार आपण त्याला म्हटलेले आहेत. सो ग्रोथ इन नथिंग बट द डार्क लाईन अँड द रिमेनिंग टिश्यू इज नथिंग बट द अर्ली वूड, त्याला स्प्रिंग वूड आपण म्हटलेलं आहे. इट इज अ लाइटर इन कलर अँड लोअर इन डेन्सिटी. त्याची जी घनता आहे ती इथं आपल्याला कमी बघायला मिळते. सो इट इज लाइटर इन कलर. हिअर ऑल्सो यू कॅन फाइंड अ लाइटर बेल्ट अँड द डार्कर लाइन्स. The spring wood consists of the larger and wider xylem elements, and because of that, it can become a darker in color. Here you can find two red pine and red oak. Two examples are given here. These are the growth rings. Darker lines are nothing but the growth rings, and the lighter lines are of the spring wood. So here you can find these two examples are given here, where you can actually find the development of the ring. You can count the rings here. So counting the ring is nothing but the counting the age of that particular plant. The spring wood is the lighter in color, and the later wood, it consists the atom wood त्याला आपण म्हणतो आपण lighter wood. That darker wood in color, it is autumn wood we can say, and having the more density of the xylem fibers. Normally these rings are compactly arranged, and the narrower elements which are have compactly developed thicker walls. Now these cells, xylem cells, when they are more compact, gives the more stronger wood, and म्हणजे that they are developing the autumn wood now. In autumn wood, tracheids and fibers are more abundant, and those found in the comparatively in the spring wood. आता याची जी ऑटम वूड मध्ये काय ट्रकेट्स आणि फायबर्स आर देअर नंबर इज लार्जर, म्हणजे झायलेम एलिमेंट्स तिथं जास्त आहेत, अँड बिकॉज ऑफ दॅट दीज नंबर इज हेल्प इन द डेव्हलपिंग स्ट्रॉंगर वूड. जिथं सेकंडरी फ्लोएम एलिमेंट जास्त असतील, नॅचरली दॅट वूड बिकम्स द सॉफ्टर वूड. Each year's growth is quite distinct. The number of annual rings correspond to the age of that particular part of the stem. आता इथं एक गोष्ट आपल्याला लक्षात घेण्यासारखी आहे की इथं आपण क्लिअर कट काय म्हटलेलं आहे, पार्ट ऑफ द स्टेम. म्हणजे एखाद्या झाडाचा बुंधा जर आपण घेतला, अ मेन स्टेम, तर त्याच्यावरच्या ग्रोथ रिंग कॅल्क्युलेट केल्या तर यू कॅन कॅल्क्युलेट द एज ऑफ दॅट प्लांट. पण कुठली तरी एखादी फांदी जर तुम्ही घेतलेली असेल तर त्या फांदीवर ज्या ग्रोथ रिंग डेव्हलप झालेल्या असतील तर ती फांदी जेव्हापासून तयार झालेली असेल तर त्या फांदीच्या वयानुसार त्याच्यावर ग्रोथ रिंग्स डेव्हलप झालेल्या दिसतील. Dendrochronology is the science where we can count and analyze the annual growth rings of the trees. आता इथं आपल्याला हे डेंड्रोक्रॉनॉलॉजी मध्ये काय त्याचं झाडांचं जर एज आपल्याला काढायचं असेल ऑन द बेसिस ऑफ द रिंग्स, हाऊ मच रिंग्स दे हॅव डेव्हलप्ड, ऑन द बेसिस ऑफ दॅट दे कॅन कॅल्क्युलेट द एज ऑफ दॅट पर्टिक्युलर प्लांट. हिअर यू कॅन फाइंड देर इज द डेव्हलपमेंट ऑफ अ सेकंडरी कॉर्क कॅम्बियम अँड ऑल दोज थिंग्स. दिस इज द सेक्शन ऑफ दॅट पर्टिक्युलर वूड. द नॅचरल वूड एखादा टीएस जर तुम्हाला बघायचा असेल तर तो अशा पद्धतीचा दिसेल आपल्याला. But when we enlarge that particular portion, you can clearly identify the outermost layer is the periderm. Then periderm is followed by the cork cambium. झाडाची जी बार्क म्हणतो आपण ब्राऊन कलरची, that is the cork cambium. Then there is the development of the secondary phloem and secondary xylem elements. So all these structures are the secondary xylems. And outer side, here it is the secondary phloem patches, and in between that there is a vascular cambium. So it is for 1 year. Now this type of the process is continuous for the multiple years. आणि म्हणून एका वर्षाची एक रिंग जर असेल तर अशा अनेक रिंग ग्रॅज्युअली डेव्हलप होत जातात. अँड बिकॉज ऑफ दॅट वी कॅन से इट इज अ annual ring of that particular plant. Then there is one category in the secondary development of the sunflower stem. Here we can found the softwood and hardwood development. It is not in particular sunflower stem, but in gradual structure we can find that there is the development secondary growth mode. We can study that. So in secondary growth in the plant, we can find the development of softwood and hardwood. Now what is the softwood? Softwood is nothing but the technical name of the gymnospermous wood, where we can find there is absence of the vessels. And because of absence of vessels, naturally there are the another tissue where the rays and tracheids are maximum in number, and because of that the wood becomes very soft because there is no xylem elements, stronger xylem elements have been developed. So it is actually not soft, but it can workable wood. The softness depend on the contents of the fibers and the vascular rays. 90 to 95% of wood is made up of the tracheids and fibers, and because of that it can be considered as a softwood. Softwood made you can find that these annual rings are more prominent. But whereas in the hardwood is nothing but the typical dicot root, where we can find there is the abundant number of the vessels are there, and because of the presence of vessels the hardwood becomes the less pore. Okay? It becomes more stronger wood, and म्हणून hardwood is normally used for the stronger furnitures, and softwood is normally used for the preparation of pencils, bats, etc., where the softwood is necessary. So this is about the not sunflower stem, but the softwood and hardwood. Then in secondary growth, we also go for the sapwood and heartwood. These two terms are there. Now when a tree is there, in our region we find there are so many sandalwood trees are there. Sandalwood tree मध्ये आपल्याला अशा पद्धतीचे दोन पॅचेस दिसतात. When we cut that particular stem, the center wood is slightly darker in color, and that dark patch is having the more value in the market because it has the more sandalwood oil, and the outer portion is lighter in color, and that has the less value. So here this outer ring of the lighter color, which indicates the sapwood or having the less amount of the oils in that. Otherwise the central wood is nothing but the having the stronger wood in the center. So these are nothing but the sapwood and the hardwood structures in that particular plant. Formation of periderm is taken place in the secondary tissue, where dicot stems produce normally a cork cambium or the phelogen in the outer cortical cells. Now these phelogen cells divides on the both and outer walls as well as the inner side, and bipolar type of the division is there to form the secondary tissue. Here you can find these are the pheloderm tissue, phelogen tissue, and formation of the cork cambium has been started here. Now here this phloem and phelogen are required to develop the secondary tissue as a cork tissue. This secondary tissue produced on the inner side is the phelogen, and this parenchymatous or it may be the collenchymatous tissue. Now it is secondary tissue or cortex of the pheloderm. It called the radial type of the arrangement of that particular tissue. The phelogen produces cork phloem, and this cork phloem, where we can say the outer side of the cork cells contain the tannins, and because of that the bark is developed on the outer side is darker in color. Hence they appear brown or dark brown in color. So it is the bark of that particular which is normally the cork tissue. And the pheloderm, phelogen and the phloem together constitutes the outermost wall of that is the cork tissue. The lenticels are typically developed in the secondary growth in the dicot plant, where we can find the lenticel is commonly produced beneath the former stomata or stoma of the epidermis, because it is the opening from where it can be tissue. So these lenticels are nothing but the openings of the tissue. Normally it is a margin raised and is formed surrounding cork cells. The lenticels are filled up by the loosely arranged thin-walled, rounded and unsuberized cells called the complementary cells. Here you can find these are the complementary cells has been developed here. So these complementary cells are loosely arranged. आणि म्हणून बाहेरचा जो झाडाचा सालीचा भाग जो असतो, so that is gradually get out of that. So it is the phloem and pheloderm tissue which can be gradually converted into a cork tissue. They enclose the intercellular spaces and it is for the gaseous section. The complementary cells are formed due to the loosely arranged phelogen and the pheloderm cells. So these are the lenticels which are used by the plant for the gradual development of the cork cambium there. Then what is the secondary growth and its significance? As we know, though, secondary growth adds to the girth of the plant. झाडाची जी रुंदी वाढत असते ती secondary growth मुळे वाढते. It provides support to the increasing weight of the aerial growth. झाड जेवढं उंच जातं त्याचं वजन वाढत असतं, and gradually it gives the support to such type of the continuous growth or in the height. The secondary growth produces a corky bark around the tree trunk which protects the interior from the abrasion, heat, cold and the infections. आता तुम्ही आपण बरेच वेळेस बघतो की झाडाखाली जेव्हा एखाद्या वेळेस कचरा आपण जाळतो, नॅचरली व्हॉट विल हॅपन, त्याची पानं सगळी जी आहेत ती जळून जातात. बट स्टील ॲट द सेम टाइम आफ्टर गिव्हिंग watering that particular plant, again the new leaves appear on that particular plant. म्हणजे जर छोटी मोठी आग असेल किंवा हीट टेम्परेचर असेल तर अशा वेळेस झाडाला इजा न पोहोचू देण्याचं काम कशाचं आहे, त्या कॉर्की बार्कचं आहे. म्हणजे आपण झाडावर इतक्या वेळेस चढतो पण झाडाला काही इजा होते का नाही, बिकॉज द बार्क इज देअर. त्याचं जे काही आहे आपल्या जाण्या येण्यानं उतरण्यानं जी काही त्याला जखम होणार आहे तर दॅट corky bark can be removed, but actual there is no harm to particular tissue of the water conducting tissue, living tissue, that is the phloem inside. It adds new conducting tissues for the replacing old and non-functioning ones. Continuous secondary growth चं काम काय आहे की जे non-functioning tissues आहेत, so that tissues can be replaced by the secondary tissues. It may be secondary xylem or the secondary phloem. सेकंडरी ग्रोथ ही प्रामुख्याने कशामध्ये होते? इट इज नॉर्मली ऑब्झर्व इन द डायकॉट स्टॅम्स, पर्टिक्युलरली डायकॉट स्टेम्स. आता हे डायकॉट स्टेम जे आहेत, in monocot also we can find there is the secondary growth is taken place. So this type of the secondary growth is considered as the abnormal anomalous growth, secondary growth, and it can be found in the arborescent monocots. आता त्यासाठी एक्झाम्पल दिले, ड्रासिना आहे, युका आहे, अगेव आहे. सो इट इज फॉर द स्टोरेज रूट्स इन दॅट, जसं बीट आहे, कॅरेट आहे, याच्यामध्ये आपल्याला ही अॅनामेलस सेकंडरी ग्रोथ बघायला मिळते, विच इज फॉर द पर्टिक्युलर पर्पज. मोनोकॉट्स मध्ये आपल्याला सेकंडरी ग्रोथ मिळत नाही, बट जे अर्बोरेसंट, जे ट्री हॅबिट असलेले आहे, जसं नारळाचं झाड आहे, स्टील इट इज इन मोनोकॉट बट इट इज अ ट्री लाईक प्लांट. अशा प्लांटमध्ये आपल्याला ही अॅनामेलस सेकंडरी ग्रोथ दिसते. द अर्बोरेसंट मोनोकॉट स्टेम्स, the secondary cambium grows hypodermal region and later forms the conjunctive tissue and the patches of the meristematic cells. आणि त्याच्यामध्ये एक विशिष्ट पद्धती सेकंडरी ग्रोथ ही डेव्हलप आपल्याला झालेली दिसते. Anomalous vascular bundles occur in the cortex, cortical bundles. Then what is the importance of secondary growth? It is mean for the replacement of the non-functional tissue which may be present in between the active tissue. So this non-functioning, non-active tissue is replaced through the secondary growth in the plants. The plants showing the secondary growth and that can be have longer compared to the other plants. Secondary growth ज्या प्लांट मध्ये आहेत तर त्यांचा लाईफ स्पॅन जो आहे तो इतर प्लांट पेक्षा वेगळा असतो, ज्यांच्यामध्ये सेकंडरी ग्रोथ नाही. It provides a fireproof, insect proof and insulating cover in the form of cork tissue. Okay? Commercial cork is produced from the secondary growth. This commercial cork is produced from this secondary growth. The wood is very important because of the secondary growth and it represents the secondary xylem.